This article is the third installment in the series Pharmaceutical Quality Risk Management. The second component in the risk analysis process is estimating the probability the process parameter or material attribute will adversely impact the critical quality attributes of the drug product or drug substance. If you have not done so, read my previous article on assessing severity, Pharmaceutical Quality Risk Management, Secrets to Assessing Severity as Easy as 1, 2, 3. With respect to assessing probability, it is only necessary to assess the probability of the variability that can impact the CQA. In this case, those parameters with the severity 2 or greater. The risk assessment process acts like a filter, only letting pertinent variability go forward in the analysis. At each stage, the process gets more detailed and focused on fewer variables. Estimating probability is more involved than assessing severity. The dataset determines the probability estimation and impacts the confidence one places on the result. This stage of the risk assessment takes process robustness into account. Process robustness is directly related to process capability. The problem with process capability is it takes a statistically valid number of runs to make a meaningful determination. In addition, process capability assumes a normal distribution of the data and the absence of special cause variation. In the pharmaceutical development phase and even immediately post-commercialization, it is hard to be certain to meet those criteria. What is needed is a way to estimate the probability of failure before getting a statistically valid number of batches. Fortunately, there is a practical method of accomplishing this. One does not need many runs to tease out the variability that impacts the process. Most firms conduct experimental design and push the process parameters to anticipated worst-case conditions. For example, API development utilizes worst-case testing by means of impurity spiking experiments to measure the purging effectiveness of the process. Many times, the purification process can be more robust in removing impurities than the reaction can generate. In such instances, the impact of the process variation is negligible. This article will not go into further details of DOE or worst-case testing. Plenty of better experts have extensively written on those topics. This article will apply the output from those exercises to the risk management process. At this point, it is important to remember the difference between correlative knowledge and causal knowledge is having a robust data set upon which to base the conclusions. In this case, the data set is comprising experimental challenges rather than extensive runtime in the plant. To understand more about correlative and causal knowledge, read my article, Quality Risk Assessment, How to Assess Risk with Limited Data. The essence of quality by design is to develop robust processes that are low risk. With respect to the highest probability, the variation that can realistically go outside the proven range or design space is essential to identify. This variability has several components. It has to do with the design margin within the operating window. Variability is a composite of the instrument measurement error and the ability to maintain process control. Inputs to these determinations come from the equipment qualification. Understanding the measurement error is an important consideration, and especially the instrument resolution. Believe it or not, I encounter many instances where the resolution of the instrument is not adequate for the required measurement. For instance, I have seen level indicators marked off in 10-liter increments, used to charge reagents to the nearest kilogram, mass flow meters, gas flow meters, and scales, each lacking the resolution needed for process control. Process development scientists must take the capability of the facility into account when designing processes. Too many times, the process design is too narrow. For example, I would typically not take a process into my API facility that had a temperature operating tolerance of ±3 degrees. Of course, the development scientist would object. I explained the calibration error of the temperature reading was ±1 degrees and consumed 33% of the proposed normal operating range. Heating and cooling control could easily throw the process into a deviation. The minimum I would accept was a proven acceptable range of ±5 degrees and preferred a ±10 degrees. Scientific justification of the process need was required if an unusually low operating window was proposed. Otherwise, the scientist should go back and widen the window. Sometimes it is not possible to widen the operating window, and one must fit into the tight squeeze. One such instance was the dry heat terminal sterilization of a drug-device combination product. The temperature and time needed to be high enough to get sterilization assurance, but also low and short enough to stop drug substance degradation. Thus, the requirements of two CQAs were in opposition to each other. There was no wiggle room. The process control, process validation, and ongoing continuous process verification plan reflected the high risk. It has been my experience that while these two dueling requirements occur, they are not the norm. The following steps in the risk management process deal with these high probability situations by utilizing detectability and implementing risk mitigation measures. Fortunately, most parameters do not fall into the very high or high probability category. The vast majority are medium or low. Higher probability scenarios are when the NOR and PAR are close to the set point and anticipated normal variability would likely or possibly move outside the ranges. Notice I added anticipated normal variability. This is not a high probability just because the operating range is low if the process control is very good. For example, the blending operation in drug product manufacture is often very well controlled with respect to blending time and number of rotations. It is often unnecessary to have wide proven ranges to increase robustness. However, most operations benefit from wider PAR or design space. The more robust the process, the lower the probability of failure becomes. I am often asked, how far do we need to move out the PAR? The answer is either where it makes no sense to increase robustness because failure is highly unlikely, or you hit the edge of failure. When the robustness is good enough that one reaches the point of diminishing returns, stop and spend effort where it makes more sense. From a process risk perspective, I prefer statistical process control over statistical quality control. SPC measures the variability of the process parameters as the manufacturing proceeds. SQC measures the variability of the output after the manufacturing is completed. Both are important, but SPC allows for adjustments to ensure the process is satisfactory. The robustness measures discussed thus far have been processing control rather than quality control. In-process control is testing conducted during manufacturing used to adjust or steer the process. IPCs are a very important control tool and will be discussed during the detectability portion of the risk assessment process. Subscribe to our channel and click the notification bell.